I'll turn around. We still need to send out the link. Okay, so now it's live. Do we think?
Just this layer.
That's probably the streaming link somehow. Miri defense. So that's good. So that's a link. But how do I go back to this? Do you know how to go back to that screen we had before? Maybe you can try. So that's where we're watching it. But somehow we're also streaming it. Is it? Oh yeah, that's fine. Great. Okay. So do I alt tab? Command tab? Oh. I don't know. Go back to somewhere we need to copy this and send it out. Okay, in fact, since we're logged into our Gmail, we can hijack her account. Yeah. Great. Yeah, you got to say, I guess figure out the email that we should send out to everybody and say, here's the updated live streaming link. And we're good. That was restful. I think it was from like an hour ago and resolved it. Yeah, that's right. That'll do. So forward, I guess. But well, yeah. Then, I think we did it. Oh yeah. Well, yeah, whatever. So I'm gonna
Do you guys want me to further up a bit?
I think we're good. Yeah.
I'll get in the middle here. Is that like nice space by you? Yeah. I'm just gonna. I'll tell everybody.
That's probably a good idea. Yeah. That's what I think.
Yeah, do you want to take that?
Okay, that seems pretty reasonable. And we can still see her.
You can open your computer.
What's that? Yeah. The links will be pretty much.
Well, actually, let's leave one over.
I think a little bit closer wouldn't be a great idea.
Yeah.
I think we'll still be able to see both, and we won't have so much of the crowd in the way. Like that, where my sandwich is.
Okay, cool. Yeah.
Yeah, I think. Thank you. Yeah, no.
And so, like, in some ways, you can make the argument that being a company like that, not constrained by publishing, study things, yeah.
I pointed out to her, she told us everything she found. This would take 3 hours, and I can't sit still for 3 hours. I can barely sit still for an hour, so hence, this would be
Great, thanks, Michael. Thanks everybody for coming. Really excited to tell you about some of the work I've done during. So broadly speaking, I've worked on 3 different areas during the time here. The first area was looking at how DNA folds up inside the cell, so to understand how the genome folds in nuclei. That was project 1, and I did that in Aaron's group. The second project that I worked on was looking at self-assembly, trying to understand what kind of structures can be built using self-assembly, which I'll explain. And the third project, which unfortunately I don't really have time to really get into, is a project I did also with Michael's group with Lucy Colo, looking at structure in noisy data sets, trying to understand how we can find a signal when the data is very noisy, and how we can use prior information to help I'm not really going to talk about that, but just thought I'd put it out there. But for now, I'm just going to talk about the first two, and specifically how structures are made inside in biology and inside cells. So, as a quick motivation, about 10 or so years ago, I was home for the summer, and my brother Rafi and I decided that we were going to take apart and build a car. We managed to get this like 1988 Nissan Sentra, tow it to the house, like dump it in our backyard, much to my parents' dismay, and we took it all apart. And that part was great. I really liked doing that. But then came phase two, which was putting it all together. And it was at this point that I abandoned poor Rafi because I quickly discovered that assembling something as complicated as a car is really no fun. You get these horrible assembly manuals, and they have all these parts that have to go together in a specific order, and you can't really wing it, right? You can't really just hope it all works out. In particular, you can't just throw all these pieces together, shake it, and hope a car emerges, right? You really have to be very careful about the order and specific ways you put things together. So the hope of having like all the car parts thrown together and a car emerging, it seems magical, but the inspiration actually comes from biology, right? So consider, for example, the ribosome. The ribosome is made up of 80 proteins, some large RNAs, and there's all these little parts, and there's nobody directing the ribosome part saying, okay, you go here, now you come here, right? Somehow, in this big jumble inside of a cell, they're able to assemble into the structure, which is known as ribosome. And so one important question is how is biology able to achieve this? And maybe we can learn something from it, and maybe one day we can assemble cars in this way. In particular, it looks like one of the important mechanisms by which biology achieves this is through specific interactions. So we have different proteins, and each protein knows that it likes to hang out with or likes to bind with other proteins, and maybe something like this can explain how these structures can. So today I'm going to talk about first some specific self-assembled biological structures that I looked at in the genome and tell you about some new structures that we found there. And then I'm going to zoom out a little bit and just talk about more general theory of how to think about these types of structures. Okay, so to start off, how the genome folds. So as a background, as many of you probably know, the genome inside every human cell is actually really long. So if you take it out and stretch it end to end, it would be about 2 meters long. Right? But somehow your genome folds up inside your nucleus, which is only 6 microns long. How the genome achieves this, huge compaction is kind of mysterious, but very interesting. So when I was a sophomore in college, I took an introductory biology class, and I got a picture that's somewhat like this. So it told me that DNA was folded at all these different levels. So at the smallest scale, at the base pair scale, I was told that there's a double helix. That of course you all know. And at the largest scales, of course, we know that DNA is organized in chromosomes. But actually, it turns out that the stuff in the middle here is actually not really well known. There's actually a lot of questions about what's happening on these size scales. Here, MB means million base pairs. Okay, so what did we actually know about the structure in these middle length scales before I started watching? So we knew, of course, that it was not completely disordered. It's not just like a random polymer packed in. We also knew it wasn't completely ordered. So it's not like a protein which folds the same way every time and has this really nice structure. You have somewhere in the middle of this continuum, but there's a lot of things that can live inside. So I'm going to tell you about one structure that people thought should exist. In the 80s, people realized that there were genes, of course, things that before, but also these things called enhancers. So enhancers, they're not making gene products themselves. They're just this element on the genome. But somehow, even though they can be very, very far away from the gene, let's say 1 million base pairs apart, somehow it was known that these enhancers are actually affecting the gene. They're allowing the gene to turn on, to create mRNAs and proteins. This is kind of weird, right? Like you wouldn't expect the switch for a gene to be, very far away. It's not like an action at a distance kind of thing, right? So from that, it was proposed that maybe these are actually close together in space. And the enhancer is actually brought next to the gene, and this allows the gene to turn on and make transcripts. So there was some evidence for this in the genome, but there was a lot of questions. So first of all, it was hypothesized that there were 1 million of these loops. It wasn't totally known where they were. So even if you know where all these red enhancers are in the genome, it wasn't clear which enhancers were looping to which gene. So that was still quite unknown. And it wasn't really known how they were made. So there's a lot of big questions about these loops, and this is one of the things that I was interested in studying when starting this project. So how do we actually go about looking for loops in the genome? It turns out that the size scales are really small. It's really hard to see DNA in a microscope. So we had to go about doing it in a really roundabout way. And the way that we did that was a method that's called contact mapping. Now this is a bit of a weird method, or I shouldn't say weird, but different. So I'm just going to give a brief analogy so people can understand the type of matrices that I'm going to show, because I'm going to show a lot of them, and it's just good to have a good background. So, okay, indulge me on this. Let's say I was interested in understanding the social structure of The Simpsons, let's say Homer Simpson. And I don't actually want to watch a TV show because, like, you know, it's okay, but not great. So instead of watching the show, I take the shortcut of going to Facebook and I start clicking through photos. Right, so I'll find photos of Homer Simpson by himself, pictures of him with Marge, with his kiddos, neighbors, and all sorts of other people in the Simpsons world. So I might get an idea of who's hanging out with whom, but I could really be more meticulous and keep a clear list of any time I saw two characters in a picture together. And once I could gather all that data, one way that I could represent that would be in what we call a contact map. So in this contact map, the columns and the rows indicate the characters in the Simpsons world, and the entry in this picture tells me how many pictures I saw those two characters together in. So immediately when you start looking at this, you start to see some really interesting structure. So the familial structure of the Simpsons immediately emerges because all the Simpsons family members are all hanging out with each other. But then beyond that, we see that Homer and Marge have a special relationship that goes kind of above the familial structure. You also notice that this matrix is symmetric, so the number of pictures that Marge is in with Homer is equal to that which Homer is in with Marge. Okay, great. So it seems like this might be a useful tool for understanding structures in the genome. The only problem is, how do we get such a map for the genome? So to do that, we use a method called HiC, the 3D genome sequencing technique, which I'll tell you about now. It was invented in 2009 by Ares' collaborators at the Broad and UMass Amherst. And what it basically does is it tries to get snapshots of the genome instead of snapshots of the Simpsons. So, for example, let's say I have two pieces of DNA, this red piece of DNA, red and blue piece of DNA, that happen to be touching in this particular cell. Maybe they're just very, very far apart on the chromosome, and I want to know that they were next to each other in this cell. So this is my method for actually gathering that data. So how do we do that? We first cross-link the cell, so this basically just freezes everything in place, so nothing's moving around. Now we cut up DNA using a biological pair of scissors called the restriction enzyme. We mark where we cut using a marker called biotin, and then we use a biological glue called the ligase, and this just glues two pieces of DNA that happen to be next to each other. So now after we clean everything up, in the end, we end up with a single piece of DNA that's actually a hybrid piece of DNA. It has a piece of DNA from the blue and a piece of DNA from the red. So now this is actually our snapshot. So if we sequence this, we can have one data point that says, oh, in one of the cells, blue was next to red. So of course, this doesn't give me much information of the structure of the whole genome, but if you sequence billions of these, then you start to build a picture of what's happening. So before I showed you the contact map of the Simpsons, the contact map of the genome looks a little different. So now the rows and the columns are indexed by locations in the genome, and this map now tells us for every location in the genome how frequently it is in contact with another location. So this map's a little hard to read. We're going to zoom in now on a single chromosome. This is chromosome 8, and this map tells me the structure of chromosome 8 inside nuclei. Okay, great. So you'll need to notice a few things that are going on in this map. First of all, there's this really bright band along the diagonal. The reason for that is actually pretty intuitive. So if you can imagine a long, shoestring, and you have 2 points on the shoestring that are near each other along the one-dimensional contour of that shoestring. When you bunch up that shoestring into a ball or whatever, then those 2 points are likely to also end up near each other in 3-dimensional space. So there's a bias for things that are near each other in 1D to have a lot of context. Okay, so a lot of other features that we see here. There's some plaid pattern, which I won't be talking about. Of course, it's symmetric. You see these white stripes here. I want you to completely ignore those because that's just areas that we don't have data for. Finally, here you see this red square equals 700. That's just the color scale. So these maps all go from white, which is 0, to the maximum value Okay, so now that we had these maps, we were really excited. We wanted to look for loops. They're really interesting, important feature of biology. But the question is, what would a loop look like in this type of map? You can sort of go through a thought exercise to figure that out. So here's my cartoon loop, and in this particular loop, I have A and B that are being pinched together. So if I think about it, what that should translate to in a contact map, if I was going to index again the rows and the columns by these little dot thingies, then what I should see is A and B should have a very strong interaction. And as you get further and further away from A and B, that interaction should just decay. So when I'm looking for a loop in a contact map, what I'm really looking for is a focal peak. The first thing that we actually did was there had been other groups that had used related methods or even orthogonal methods to find loops in the human genome, and they put out these lists for people to look at. One example of that is the ENCODE Consortium. So the ENCODE Consortium is the National Human Genome Research Initiative. They had a project to sequence and do a lot of genetic sequencing of the genome, and one of their landmark papers was to put out a list of loops that they found in the human genome. So what I did was looked at these loops on our maps and to see are there actually peaks there, or are they saying that there are. But I was really disappointed because when you zoomed in on these peaks, or on their alleged peaks, there was often not any focal enrichment. So I was pretty disappointed by this. And then we thought, okay, well maybe what's happening is these peaks are just not as enriched as I think they are. Maybe they're supposed to be twofold enriched, maybe they're just less enriched. We thought maybe some aggregate analysis of all of these peaks would help us see this structure. So to do that, I designed a simple test which is called aggregate peak analysis, which just draws a square around all of the loops. I've only drawn around some of the loops here, and then adds those matrices together. And when you get this aggregate matrix, what you get is an aggregate signal of all the loops in that list. But again, when we looked at this list, we took the aggregate signal, we did not see a peak where we should have seen one if there really was focus. So to say the least, we were very puzzled by this. So basically, this led us to want to do just make better maps, make very, very high resolution, more sequencing maps. So that's what we did. So the original maps, the high-speed maps are shown here on the left. These were from 2009, and they could see structure at these large scales. But once you got down to the very high resolution, it was really hard to see anything. These maps are just very sparse. And since then, there have been other groups that have worked on this, but the experiment that I looked at was data which had 5 billion contacts, so basically 5 billion of these snapshots, and we were able to get maps made down at these very, very, very high resolution, down to 1,000 base pair resolution. So of course, we had this great data set, and now it was just a matter of going forth and exploring and see what we could find. Here's chromosome 7. I'm going to zoom in really close to the diagonal, and lo and behold, when we did this, we were very excited to find that indeed we now saw these focal peaks, sort of as we expect, they have a very strong interaction and in this case get further away from them. So we're really excited about that, and it was just basically like, let's go find all the peaks of the human genome and figure out what they're about. To do that, we first, I should say that they're often zoomed in here with 200,000 base pairs, so this is very close to the diagonal. So to find all the peaks in the human genome, we first had to create an algorithm to do that, and we did that. It's called Pickups. This is work that was led by Sue Asrell and Eric's group. This algorithm, I won't talk too much about it, but it basically just finds all of these peaks. So we did that, and then we were able to make a catalog of the loops of the human genome and try to figure out more about them. So there's a lot of questions that you want to know about these loops, like how many are there, what do they do, how do they form, etc. So let's just tackle these one by one. We found about 10,000 loops in the human genome. My first draft of this talk, I was going to show you all 10,000 of them, but I decided I'll go easy on you and let you get subset. So here I'm showing the primary, like one experiment and the replicate experiment, and you can see these loops replicate time and time again. So we're pretty confident. They're very confident these are real. They title the human genome so that like this scale representation of what we think, or not to scale, an accurate representation of what we think a specific example in the genome looks like. And we think pretty much everything is in one of these loops. And particularly, we did not see a million loops. So we do the same analysis that I showed you earlier on these loops. When you zoom in on any loop, of course, you see a focal enrichment, and when you add all these loops together, you do see a very, very strong focal enrichment. Not just in the map that we called these peaks in, that would be expected, but also in any other high-C map that we have been published to date, we saw a strong enrichment at the center. So we're very confident in these loops. So now, of course, you're wondering, are these loops, do they happen every time in every cell? Does your eye cell have the same loops as your liver cell? So the answer was many times they do. So this is an example of one loop in different cell lengths, and you can see it's in every single one. And this happens for many loops. But there's also some exceptions to this. So there are some loops which are not conserved between cell lines. And so, for instance, I'm showing you two different cell lines here. There's a lot of structure happening in this one, and there's not so much happening in this one. And in particular, the structure here, these loops here are actually looping to a particular gene. It's called IMTS1, and this gene happens to be on in this cell. In this cell, that gene is off, and there's really not a lot of loops happening. So it seems like this looping is actually doing something to help turn these genes on. And this is a really tantalizing idea because, as many of you know, right, your eye cell and your liver cell have the same genome, right? But they somehow achieve very different functions. So it seems like this folding is somehow able to help with having different cells achieve different functions. Okay, so I think a lot of this talk is about self-assembly, and in particular, for this, we wanted to know how do these things assemble? What how can you actually form a loop? And the first thing that we looked at is what sits at these anchors. What's sitting here, pinching these two together. And we were really surprised to find that when we looked at what was hanging out exactly at these loop anchors, we saw something interesting, which was that CDCF, a particular protein, was sitting at all of these loop anchors. So we know CDCF pretty much that exception at all of these loops at both sides. So we thought that was great, but then we found something which we thought at the time was really shocking. So CDCF, it's known that it doesn't bind to anywhere in the genome. It binds in a particular motif or a word that sits in the genome. So we know where those are. But that word is not symmetric. It's CCA something. And it can be written either in the forward direction or it can be written in the backwards direction. Now, if you have two motifs, paired motifs, there's four orientations they could be in. They could be pointing towards each other, pointing to the right, the left, pointing away from each other. And a priori, you wouldn't expect these two motifs to have any orientation with each other. But we found that in pretty much all of our loops, they are actually always in this convergent orientation. You have to have these motifs facing each other for there to be a loop. So this sets a really strong, a really stringent requirement for forming loops that doesn't really make much sense on the outset, right? It's not clear why you need to have this strength requirement. But so the picture that we think is happening is, so we have words, they're both written in the same direction, and you have CDCF, this protein, which is binding to these motifs, and somehow this is the requirement for loop setup. Okay, so I keep drawing this cartoon here. It's a cartoon, right, because I haven't really talked about what's happening, in between these two pinch loci, and many of you probably already guessed what the answer is. I keep showing this image of this loop. But as you know, in between these loops, there is this increased contact frequency every minute, and that should be reminiscent of this multiple structure where everything, basically all of the loci that are hanging out in between these two anchors are also hanging out together in this segregated way. So the picture really should be something like that. One of the things that we have to do was annotate all these domains. So one of the things that I did was wrote an algorithm that first called the arrowhead algorithm, and it transforms the contact matrix into the arrowhead matrix, and then it annotates these domains genome-wide. We found about 10,000 of them. They're 200 to 300,000 base pairs long. It's how the genome. Okay, so it seems like these CDCF motifs are very important for loop formation. And one question is, do they happen to be there, or are they playing some sort of causal role? And to do this, try to analyze this, this was work that was not done by me, but by Adrian and Stuart, experiment, where he actually just cut out one of these motifs, just like deleted using CRISPR this actual motif. So this is the wild type, and you can see there's a loop anchor, these arrows indicate the orientation of the motif. And then he deleted this one. You can see immediately that both the loop, which used to be here, and the domain that used to be there, just completely disappeared. So it's clear from this that CDCF, the CDCF motifs are actually necessary for these loops to form. Okay, so just a quick summary of what we learned so far about these loops. There's about 10,000 of them. They turn genes on. They form domains in between where the loops are going to stop. And they have this very stringent requirement of a convergent CDCF orientation. Okay, so we thought we were done, and we were very happy with this. But we finally can get some sleep. But it turns out that there was a bit more to this story. And what happened with this is I was reading a paper by Brian Chadwick, who's a researcher at Florida State, and he, in this paper, they claimed that they saw long-range interactions in the inactive X chromosome. So just stepping back for a moment, female humans have two X's, males have X and Y, but females have two X's. One of those X chromosomes is on, it's making genes. The other one is actually just completely off, or for the most part off. It's called inactive, and it's just, pretty boring. It just hangs out at the corner of the nucleus and, shoved to the side, and it's not making any genes for the most part. And so they claim that they saw some long-range interactions in that foreign inactive chromosome. So we wanted to follow up on this and see if we could see those in high-C data. But, the problem is high-C data really just looks at both chromosomes together. You can't really separate out the two. So to do that, we have to come up with an algorithm to separate out the two chromosomes. And we did that. This was work that was led by Miri and Aaron's group. And looking at the mutations that between mom and dad, you can basically make contact maps for both the paternal and the maternal copies of the chromosome. This is chromosome 3. Actually, this is pretty boring. Like, both copies look very similar to each other, and that's to be expected. We expect both chromosomes to be doing the same thing. But now using this technique, we can now look to see if long-range interactions actually existed on the inactive X chromosome. So we looked, and it turns out they in fact existed. So this is the inactive X of one of the cell lines, and there was a dot exactly where this paper had to there was one. And if you look at the active X, the more exciting one, there actually was no loop. These loops exist in all female cell lines that we looked at, all stereotypical normal female cell lines that we looked at, and not in male cell lines. So you might think, oh, well, this just looks like the other loops that you were talking about. Turns out they're actually quite different. So the other loops that I was talking about had, at maximum, they were 2 megabases, but actually most of them are around 200 or 300,000 base pairs span that width. Whereas this loop that we found, and in fact we found a whole class of these loops, there's about 25 of them on the inactive X chromosome, and all of them span these really huge size scales. So the largest one is 75 million base pairs away. So it seems like these are quite different than these other ones, and not knowing what to call them, we call them super loops just because of their size scale. So it seems like this class of loops is pretty different, but one really interesting feature was if you looked at the anchors where these loops were residing, and one anchor in particular, which is called DXC4, it not only had one CDCF binding site and protein, but it actually had many, so it have hundreds, which is very suggestive, right? You only need one CDCF motif, small loop, but maybe you need many to set up these so we knew that these quote-unquote normal loops set up normal domains, and now we're wondering, what do these super loops do? Do they set up something similar? And indeed, when we looked at the whole inactive X chromosome, here shown on the right, it had these very large domains split exactly at this genetic element at DXC4, which had these CDCF binding sites. Obviously, this is very different than what we saw in active X, which did not have these super domains, nor did it have super loops. So since since we had seen that if you delete for normal loops, if you delete the CDCF motif, you can destroy the loop and destroy the domain. We thought, well, what happens if you delete not just a single motif, but delete all of them in this particular locus? So we did this. Emily Darrow and Brian Chapman's group made a Killen solution of DXE4, about 100 QB deletion. And then I did the high-C experiments to compare that to a wild type. And indeed, once you do this deletion, this structure goes away. So the domain goes away and the superloops go away. So this indicates that the CDCF anchors are important not just for local loops, but also for the superloops and superstructure. Okay, now going further, we were interested in is this an important feature, these superloops, super domains? And one way to understand if something is important in biology is to see if it has been conserved at all through evolution. Humans invented it, maybe it's just a one-off thing, maybe it's not so important. So I did some experiments, some high-C experiments in mouse, and found that indeed these superloops, which were found in human, also exist in mouse and only on the inactive, not on the active. And similarly to superdomains, split at where DXE4 is in mouse, which is relatively human. But we also found the superdomains in the inactive mouse and not the active. So this indicates that the superstructure, it suggests that the superstructure might be important for exit activation since it exists across at least a few species. Okay, so again, to sum up, we've now talked about two classes of loops. These normal loops, which are much smaller relative to super loops, and the super loops as opposed to normal loops have only 5 of them, only on the inactive X. They're associated with these with super domains. They have some of the anchors have many CDCF sites, and if you delete the CDCF, you disrupt that structure. Okay, so stepping back for a moment, one question that emerges through all of this, especially given the themes of this, is how do these structures actually form? How do these loops actually assemble inside the genome? And I just briefly want to show some work that I did, work that others in the group did and I worked in collaboration with, coming up with the model to explain this. So this is this is a movie from Adrian Sanborn and Suas Rao's recent paper. Hypothesizing the existence of what they call the extrusion complex, which looks a little bit like a double donut, but it lands, the idea is that it lands on DNA, and DNA is then looped through these donuts as the extrusion complex moves down the chromatin fiber. So then the question, okay, that would maybe explain how loops form, but how does it actually stop? So the idea is that this extrusion complex stops once it hits the CPCF motif, but that motif has to be pointed in the forward direction. If it hits one that's in the backwards direction, it just keeps going. Only when it hits a motif in the forward direction would it stop. So that's the idea of the extrusion complex. And molecular dynamic simulations, looking at this mechanism, are able to explain the data very well. So this is an experiment, and these are Adrian's molecular dynamic simulation. All of these simulations get as input is just these motifs, and then we get on them, and we're able to recapitulate these feedback. And recently with Adrian, we found that simulations, if you give this model the CTCF binding for an entire chromosome, for the X chromosome, you can actually recapitulate the superdomains and the superloops. So it seems like there is just, we're able to get these types of structures through self-assembly through extrusion. Okay, all right, so I want to now shift gears a little bit and talk more about, more generally, how these types of structures, whether it's extrusion or whether it's ribosome assembly, how they can be made. And one key part of everything that I've been telling you with regards to these groups is CDCF binds to a particular word found in DNA. And when you think about it, it's kind of strange. How does this protein know that it likes this particular word? And how can we use it to build things? So proteins and biomolecules in general use basically two different mechanisms in combination to recognize each other. So the first is that of shape. So if two proteins are meant to bind, oftentimes you'll have complementary shapes that encourages them to stick together. But usually that's not all, right? Usually there is some type of surface chemistry that happens on top of the shape. So for instance, if I have hydrophobic, hydrophilic coating of my protein, that encourages these two shapes to come together. And so proteins really use a combination of these two things to do everything they have to do. And these interactions, which we'll be calling here specific interactions, are really the building block for a lot of things in biology. So for instance, you can imagine once you have a set of specific interactions, you could do something like self-assembly by combining them in certain ways. But you can also do all sorts of other things. You know, for instance, you could do cell signaling if you require two proteins to bind for your capsid downstream signal. This is really a very general tool for protein recognition. And here's an example of two proteins which are meant to bind. One is concave, one is convex, but they also have this electrostatic coloring on their surface, which encourages them to bind together. So it would be nice if we could do this kind of stuff, you know, to build a car. So people have been working on experiments to try to do this. So many experiments look at shape. So they'll make colloidal particles on a micron scale of two shapes, like for instance this football, which is supposed to bind to its Pac-Man partner. Other experiments use surface chemistry to try to do this. So some of them will take two surfaces and they'll paint down hydrophobic and hydrophilic paintings, and you know when the two surfaces come together, they'll register. Many other experiments use DNA. So if you have two pieces of DNA that are complementary to each other, they'll bind, and we can use it to build larger structures. And so some of these experiments have been able to build really cool three-dimensional structures using this technique, which is great. But I think that, you know, if the ribosome saw this, it might like pat us on the head and be like, okay, you know, good job, right? Like we're still not quite where biology is in terms of being able to settle these kinds of things. And so I was really interested in understanding why this is, like what sets the limit, how complicated the structure you can build using specific interactions. So, you know, in particular for these cartoon pictures that I've been drawing, one might ask, you know, how many different ways can I draw this shape? How many different distinct shapes can I actually have? In particular, for instance, is this, you know, are these two eggshells different than these two? It's not totally clear just looking at them whether or not these are distinct entities if they were hanging around in a cell. So, for instance, let's say I had, you know, 4 components or component pairs hanging around the cell, and each is supposed to bind to a specific partner with some fidelity. Now, if I add this other pair, suddenly my interactions get really messed up. So I have all this off-target binding where, you know, this one binds to this one and this one binds to this one, even though they're not really meant to. And so somehow we want to say that this is bad, like this should not be good for your system, but we need to be able to quantify this and understand you know, how this affects the structure that can be made using this type of system. And just to go back to the previous analogy, you know, if you were drawing Simpsons characters, one question you might ask is how many distinct characters can you actually draw? You know, how many different princes that you draw these yellow, blue-haired, or it's not just blue-haired, but you know, goats. And you know, for instance, Marge has these sisters which are identical twins, and even though they're distinct characters on the show, for the most part, they're actually treated identically. So if you draw them in this contact matrix, you can actually find that their 2 rows are actually identical, and their columns are identical because, you know, they're pretty much always together. And you also see a lot of, you know, off-target interaction because they're always together. They're always together. So, but just to bring this back to protein land, we want to know again what are the limits of the type of system that you build using these types of interactions. So this project, this is the project I worked on with Arvin Murgan and Michael's group. We actually really lucked out because it turns out that Shannon, something like 50, 60 years ago, actually worked on a problem that was related, but in an orthogonal problem, really. So what he was looking at is communication along a channel. So he was looking at telegraphs and wanted to know how much information can I send along a channel. And what he found was something really interesting. When he calculated what he called the mutual information, which I'll explain, when he calculated that as a function of the number of messages that he was sending along this channel, I want to send like 10 messages per second or 20 messages per second. So if you plotted the amount of information that was actually getting through, he found that that information actually reached a distinct maximum, and that there was no point in sending more messages than is indicated by this maximum. So we call this maximum in mutual information. We call this the capacity of the system, and this suggests the maximum number of messages that you should use for this channel. Like I said, we were actually really lucky because it turns out that we could really just translate this framework into our framework of specific interaction. And the analogy worked really well. And it turns out that if you plot the mutual information for specific interaction components, we find that again, we have this maximum in the mutual information, which suggests the capacity of the system. And it also suggests the maximum number of components that you should use in your system. Okay, so capacity is a useful tool for describing these systems. We'll show you a little bit of how we calculate it. So mutual information between two sets x and y is defined with the following equation. And in particular, it looks at, for our case, it looks at the probability that x binds to y. So how do we get those probabilities? Well, these matrices that I'm showing you here are probability matrices, which tell you what's the probability that, Pac-Man binds with this eggshell, for example. And we actually can figure out what that probability is based on equilibrium statistics. In this model, the probability that these two components will bind will just be proportional to a Boltzmann factor of their energy. Okay, so now we can talk about mutual information now in terms of energy, and now I'm going to define a number or a parameter called delta, and this is going to be the crosstalk gap. So this just represents the difference between on-target binding and off-target binding. And I guess the idea here is that if delta is very small, if this difference is very small, then there's a lot of off-target interactions and a lot of crosstalk. If delta is very large, then you don't have any off-target interactions, and it's somehow better, right? So why is this useful? It turns out you can abstract away all these details and just say for a moment, let's say I don't know exactly what components are in a mixture, but I do know that this crosstalk gap comes from a distribution called rho of delta. So it's some distribution of crosstalk, maybe it's a Gaussian, maybe it has a weird shape, etc. It turns out that you can calculate the capacity as a very, very simple function of this distribution. And in particular, the capacity can be approximated as just the log of an integral of this distribution. And this integral is basically a measure of delta, of the crosstalk gap. And so capacity increases the bigger the gap gets. So the harder it is for two off-target components to bind, the larger your capacity is. So this really corresponds to our intuition. But more than that, it now allows us to calculate. So if I'm given a distribution, for instance, this Gaussian distribution, I can now easily calculate what its information capacity is. And in this case, it's 2.4 bits, and it suggests that it should be 111. Now, if we look at these two other distributions that we have on here, they're shifted to the right, and so that means larger delta, so we expect the capacity to be bigger. And indeed they are, and the capacity of 2.6 bits. But the interesting thing in this case is actually these two distributions have the same capacity, and that's because I just cooked them up such that they had the same integral here. But the important takeaway from the slide is really that you can calculate capacity easily once you know the distribution of the crosstalk energies. And there's lots of systems that you know the crosstalk energies for, and so you can just take your favorite system and then just ask what's the capacity. So we went and did this, and in particular, we were interested in understanding how the capacity changes as a function of various things in the system, temperature or ionic charge, etc. So now I'm going to briefly talk about just shape. I know I previously mentioned shape and color, but this is a model which looks at the binding of two shape components. So here's component one. Here's its complementary pair. They're supposed to stick together. And this interaction here is mediated by the depletion force. These are supposed to be depleted particles. And this is not really important for the general concept, but the way the depletion force works is that you have a bunch of particles and they for when these two shapes are stuck together, because there's more entropically favorable, because there's more configurations for them to be when those two shapes are stuck together than when they're far apart. So it encourages any two surfaces to stick together, non-specifically. So this is your on-target pair, and the energy that you get from binding is just proportional to the contour of their surface areas. The off-target pair would look something like this. They really don't touch in any places, and so the difference between the energy between these two would represent the crosspath gap for this. So now we want to know how capacity scales with all sorts of things having to do with model. One interesting thing that we looked at was looking at the length scales. So there's two relevant length scales in the y direction. One is delta, that sets the height of these bumps, and one is d, which is the height of these depleting particles. And using those, we can define a single dimensionless parameter here, delta over d, which we call the shape precision. So it really tells us how much the physics can really read of this shape. So if d, the depletion particle size, is very, very, very large, it's actually not going to be able to tell the difference between a small bump and a small bump plus a one, right? Whereas if the depleting particle is small, it has it's able to really recognize every little nook and cranny in the shape. So we expect that the capacity should increase the larger delta over d gets, and now we can actually calculate it and quantify this effect. And indeed, we find that it increases exponentially with it, which is something we would expect. Yes.
Can you explain a little bit more about why the depleting particle is important to the physics of this?
So it's actually not. What's important, so I guess the depleting particle here is just serving as a proxy for the length scale of force. But in this particular case, these shapes, this model is relying on the fact that two surfaces like to stick together non-specifically. So that could be van der Waals, that could be anything you like that just makes two surfaces stick together. In this particular case, I've chosen the depleting particle just because it's a little clearer, but the important thing here is that you just have a force which non-specifically wants these two things to stick together, and every force has a length scale. So in this case, the length scale that the force sees in this case is just the depleting particle size.
Can you clarify why the depleting particle creates a force or is associated with a force?
Yeah. So I think the basic idea is so if you have these depleting particles, if these two shapes are far apart, then the depleting particles can go anywhere, except they can't actually go all the way. They can't get. There's a hard wall here, right? So they can't. There's a particular area here where they along the contour that they can't actually go. But basically, there's a forbidden area that's set up by the hard wall. When you push these two together, now suddenly you've reduced that forbidden area for these depleting particles by half. And so now you have just more volume in which these depleting particles can hang out. And so one way to think about that is just entropically, there's just more ways for those depleting particles to be arranged when you switch those two surfaces together. And so that encourages them to switch together. But I think the details are really not so important for this. The important thing is that it's like surfaces to go together and it's consistent. Okay, so now we've talked about shape. I've also mentioned the surface chemistry, this color model. So I'm going to show a very basic model of that as well. So in this model, I'll have two components which bind to each other. And in this case, purple like purple, orange like orange, or maybe it's hydrophobic hydrophilic, maybe it's a different layers of DNA, etc. And the energy that I'll get from this is proportional to the length of these. Now, in my off-target binding case, I'll have two things come together. They just start, let's say they have random colors on them. So there'll be some places where they match up. And so the binding energy that I'll get penalty from this will be proportional to in this case too. So now I want to contrast them to the shape model, which I already told you about, and ask, which one's better. It's hard to say which one's better, right? Because you can just change the parameters for either one. But we can ask which one scales better as a function of different parameters of the system, and in particular, the length of these components. So for example, let's say this thing has 6 subunits. Let's say I was gonna add 2 more subunits onto it. What would happen? Well, the energy of the on-target components just scale linearly because your probability of having any two new subunits having the same color stays the same. And so, for instance, in this case, we pick another one, and that, and just in general, that amount will just scale. Okay, now what happens to shape, right? What happens if I add, two new Tetris bars to the right? Well, what happens is, my on-target binding did fine. I got, more contour on this, so that's good. It scales linearly. But my off-target binding, something actually really interesting happened. I actually didn't get any more contributions because it had already been touching in one spot, and when I added these two new bits, nothing really changed. And the intuition for this is actually pretty clear, right? If you take two very rocky rocks and you shove them together, they usually can only touch at one spot. They're not going to be contorting to touch each other in many spots. And so it turns out for the shape model, you can barely see this, but the off-target binding actually is relatively constant with respect to the length. So that was a comparison of shape and color. I told you at the beginning that proteins use not just shape or color, but some combination of the two. So you really wanted to understand, like, is there a way to comprehend why that would be advantageous? So to do that, we devised a very simple model, two simple models. So the first one is shown here. If I were to combine shape and color, the simplest way I could possibly do it would be to concatenate the two, right? Just stick a tether in between them, and so now my off-target and on-target binding is just a combination of this. And so in this particular case, my off-target binding will get a penalty from the one place where the two shapes touch, and I'll get a penalty in these three places where the colors match from these two off-target pairs. And it turns out that the capacity for this system, which is concatenated two separate components, actually just scales as the addition of the capacity of the first two. And this makes a lot of intuitive sense. If we have two channels and they're independent of each other, the information should be equal to each other. But this is not what proteins do, right? Proteins are using shape and color in conjunction. So our model for that was we took these shapes and then we painted color on top of them. So our on-target binding was green is matching with green and it's touching green. And the off-target component, though, is something interesting. So these are the exact same shapes and the exact same colors here and here. Here we get one penalty energy from where the two shapes touch, right? But there's no places where the two colors match. And that's because it's very unlikely that the one place that they'll touch or the few places that the two shapes touch, they will also have the colors matching. And so somehow these two are working in a way that's symbiotic in some way that allows you to really reduce the off-target binding in a way that you could not do just by their concatenation. And one way to look at this is to look at rho of delta, this gap energy distribution. So the gap energy distribution for these models that we showed here would be just for color, and here would be just for shape. When you concatenate them, you end up shifting to the right, and this gives you a distribution for this concatenation. This is actually the composition of these two. That's not important. The important thing is when you paint color in the way it's similar to proteins, you end up shifting it even more to the right, and so you can get a nonlinear bump in how your capacity scales, even in this case, which is just random shapes and random colors. So this suggests really that maybe these experiments that are focusing on shape and on color, maybe what we should be doing is somehow combining the two to achieve something like. Okay. So, yeah.
So, one thing that's really important in Shannon's original analysis is that you're sending a certain amount of signal over your channel in a given unit of time. And can you comment on how the shape and color potentially affect the time scale at which information can be communicated in biology?
Yeah, I think so. I guess all this entire model is at equilibrium, so there is no time. You can try to put in time if you want, and you do non-equilibrium stuff, but everything I've shown so far is non-equilibrium. So there really is no time in these models. I think the fundamental idea really is, even though Shannon was talking about how much messages you can send per unit time, the basic idea was really that you have this channel and it has a limit, has an information limit, right? And I think that's the limit that we're drawing an analogy to, just the information limit of these systems. But there's not really any time in the system. If you wanted to add time, you could, and you could, for instance, calculate the binding probability as a function of time. That's something that you know you can go ahead and do. But we didn't. Okay, that's actually it. This is a lot of stuff. Told you about genome folding, and I told you about a little bit of theory of self-assembly. And I didn't tell you about this third project, but I think the basic idea that I want you to take away is that there's lots of really cool and interesting structures in the genome, outside the genome, biology in general, and we don't fully understand them, but I think it's getting just really fun to study them and develop quantitative methods. Before I totally finish, I want to thank a few people who have been really instrumental to my activity. First, I want to thank Miri, Michael, and really want to thank both Michael and Erin for being really wonderful, great advisors throughout this whole process. They've been great mentors to me. Era's, I tried to find the most embarrassing photos. So, Eric has really been over the top, like in terms of his dedication to me and to all of the students, and he's really willing to dive into the detail, and work out any problem that I have on any aspect of my work, and the one who's really benefited tremendously from that. So I really want to thank him for his guidance and his help throughout the years. It's been really wonderful and unique experience working with Eras. You might not recognize this person as Michael Brenner. I've never seen him in a suit and tie, but it turns out he might not like me for saying this. But he was a guest judge on Top Chef a while ago. So watch that. Michael has been really wonderful as a mentor. I think the first thing I learned from Michael was AM 201, which I think a lot of you also took, where he really emphasized to us, basically kept saying, don't be scared of complicated calculations, right? Don't be scared of scary equations. And he really has continued over the course of working with him to encourage me to really have courage when it comes to doing these types of calculations, and just in general to explore different aspects. I want to thank him again for his guidance and mentorship these past few years. This work has been a presentation of work that did many people. All of it was a collaboration. So, for example, all the IC work, the cell paper was done with all these folks. It was co-led with Suhas Rao, who is a really talented researcher at Stanford, and we really worked really closely on every aspect of this paper together. I mean, they got countless nights working together. He was a really great person to have as a partner on this project. And I also worked closely with Neva, and she's taught me a great many things, especially with regards to coding, and Elena, who was very patient teaching me all the IC experiments, despite my really lack of ability in that area. I want to thank all the other members of the Aiden Lab, Emily, and Brian's group who worked for the DX4 division. They've been great. And Adrian, I've been working on with all the extrusion for extrusion. I've also had the great fortune of working with many of you in this room, in the Brenner group, and also the Pierce Hall family. So the self-assembly stuff was done with Arvind, and just has a really delightful way of looking at science. That's really fun to work with. Lucy Colwell, I worked on the random matrix theory project, and she's been great. She's taught me so much, especially about proteins and about life in general. And all of the folks who have really been, it's just been great to have discussions with all of you. I guess I mostly just presenting my academic work, but my life in graduate school has been really tremendously just made so much better by the community that I've been part of. So Nashton has been really wonderful, and I couldn't list all of the roommates that I've had over the years, but here's some of them. And just in general, just Graduate School. I want to thank Noam for organizing Asasa. It's by far the best journal club that could ever possibly exist, and I think everybody's been to it, I'm sure agrees. And then I just want to thank all my friends, both old and new, for their support over the years. I just want to thank John for his support and tremendously enriching my life. You're remiss not to mention my wonderful siblings, Nana, Rafi, Ariel, Michelle, and Andre. They are the goofiest people I know, and they keep me from taking myself too seriously, but they've been just great friends. And finally, I want to thank my parents who have really sacrificed so much over the years and really supported me and my siblings for so long in all their educational endeavors. And I want to thank you for your support. So that's it.
You guys want to ask questions?
I had a question of the superloops. All the CTCF domains that were sort of next to each other, were they all facing the same direction?
Good question. In the TXT4 element that I showed you, actually, they're not all the same.
So the ones that are paired, like, are they facing complementary?
So a lot of those are tandem, so they're in both directions, both of them, and yeah, but don't which one's which one. But yeah, good question. Obviously important for terms of like things like the model. Other questions?
So for your contact model, have you explored a little bit of flexible contacts? I guess I would decrease the channel capacity, so I guess.
Yeah. Oh, okay. Yeah. Good question. Right. So I think especially like in biology, right? Like things are squishy, and there is, in fact, you may think that like two stages. The first they have to come together, and then they squish a little bit, and they come together a little more. We haven't done that, but we thought about it, and I think that would be a natural place to try to make this more akin to proteins, which are not hard, but rather are squishy. And imagine even some non-equilibrium dynamics again to try to get this time aspect in. But I mean, I think in the end, if you know, like let's say you can account for all the squishiness in your model, and you know what the distribution is, accounting for all the squishiness. Then if you can describe it as an equilibrium model, you don't actually need to care about the squishiness at all. You just now you have the distribution, and you can write the capacity of
The audience is stunned. Okay, so if not, I close your clap. Everyone has to except for who will deliver. Just listen. Unless other radical nasty.